Hi students, now we are discussing anatomy of flowering plants or anatomy of higher plants, and we can expect 2 to 3 questions in our Neet examination. Anatomy, which deals with the study of internal structure of an organism, and we know that in higher plants or higher organism, cell group to form tissues, tissues group to form organ group to form organ system, and ultimately organism. So there is a hierarchy of cellular grade or organizational grade in our all higher or multicellular organism. And the branch of science that deals with the study of tissues are called as histology. And what is tissues? Tissues is a group of cells. Tissues is a group of cells which perform a common function, and it has a common ontogeny or origin. Tissue is a group of cells which perform a common function, and it has a common structure or common ontogeny, similar origin. And the branch of science that deals with the study of tissues are called as histology. And the word histology was introduced by Bichat. The word histology was introduced by Bichat, and the father of histology is Mayer. And when we are analyzing the internal tissues, internal structure of plants, plant has mainly 2 types of tissues. Plant has mainly 2 types of tissues. One is meristematic tissue. Meristem, the word meri, which means divided or small fragments. Meristem, small pieces of cells, and permanent cell. So in the plant, tissues can be divided into 2 types, meristematic tissue and permanent tissue. And what is meristematic tissue? Meristematic tissues are present only on growing part of the plant. Meristematic tissues is present only on growing part of the plant, and they are actively dividing cells. They are with actively dividing cells. So they are present on the dividing part or growing part of the plant, and they are actively dividing. We know mitosis. Actively dividing cells. When the cell undertaking division, the cell must have well-defined, well-developed nucleus. It has a large, well-developed nucleus. That is another characteristic. Then the cell must be with abundant cytoplasm. Large quantity of cytoplasm must be present. That is another next characteristic. Then the vacuoles are absent. We know that a mature plant cell is with large vacuole. About 90% of total volume of the cell is occupied by vacuole. But in the case of meristematic tissue, vacuoles are absent. If it is present, smaller in size. So meristematic tissues are the dividing tissues, and they are present only on the growing part of the plant with different characteristics, like they are actively dividing cell. If they are undertaking active cell division, there must be well-defined nucleus, abundant cytoplasm, large vacuole, small vacuole or vacuoles may be absent, and the cell wall must be thin and composed of only cellulose. That is, no any other chemicals like hemicellulose, pectin, suberin, lignin is not present, or it is totally absent in the case of meristematic tissue. So meristematic tissues are actively dividing cells present on the different growing part of the plant. And on the basis of position in the plant body, the meristem can be divided into 3 different types: apical meristem, intercalary meristem, and lateral meristem. When we heard the term, on the we can expect what is the position. Apical meristem, it is present on the tip of the plant body. And in this tip region there is meristem, and that is called as apical meristem. And these cells, the group of cells which undergo active cell division, and that results in the growth of plant in length. So apical meristem is present in the apices of root and stem. apices of root and stem. And intercalary meristem. Apical meristem is present all plants, all angiosperm, both dicot and monocot. Apical meristem is present. But in the case of intercalary meristem, when we take a stem, this is a nodal region. This is another node, and node is a region where the leaf or branch will originate, and just beneath the nodal region there is a group of meristem and which help in the growth in length or elongation of stem, and this meristem is called as intercalary meristem, and the intercalary meristem is present only in monocots, especially grasses. The length of internode of grasses like grass plant, and also in the case of bamboo, sugarcane, everything, it is taken by due to intercalary meristem. So intercalary meristem is present in the intercalated, in between the two node. In between the two node we can see a meristem, and that meristem which help in the growth in length or internodal elongation. And the third type of meristem which is present on the lateral position. When we take the stem, it is present on the lateral side, and this type of meristem is present only in dicot plants, gymnosperm and also dicot plants, and it help in the secondary growth, that is vascular cambium and vascular cambium, intervesicular cambium and also intravesicular cambium are the example for lateral meristem. Lateral meristem which help in the growth in diameter or secondary thickening is done by lateral meristem. So on the basis of the position of meristem, it can be divided into apical meristem, intercalary meristem and lateral meristem. So apical meristem, apex or the tip of the plant body like root and stem, which has apical meristem, and it is present in both dicot and monocot. But in the case of intercalary meristem, it is present only in monocots, especially on the grasses, and lateral meristem which is present on dicot, and lateral meristem help in secondary thickening. Secondary thickening. And this is the basis of the position, and we can be classified this into these three types of meristem into two types. One is apical meristem and intercalary meristem present on the plant body from its birth. From the embryonic stage onwards, this meristem is present. So apical meristem and intercalary meristem are primary meristem. Primary meristem. Apical meristem and intercalary meristem are called as primary meristem because it is present from embryonic stage. But the lateral meristem like vascular cambium and core cambium, which develop in the later on their life, so it is called as secondary meristem. Secondary meristem. So secondary meristem is present only in dicots. Secondary meristem present only on dicots. So what is meristem? What are the characteristics of meristematic tissue? And what is the classification of meristem? Meristem is present on the maybe on the apical region or intercalary region or on lateral side of the plant. And the second type of tissue that is called as permanent tissue. And permanent tissue, they are deriving from the meristematic tissue, and they loses their ability to divide. They loses their ability to divide, and they are differentiated tissue, and they perform specified functions in the plant body. Meristematic tissue has only one function, cell division only. They are dividing and produces the permanent tissue, and the permanent tissue on the basis of their structural complexity, it can be divided into two types, simple permanent tissue and complex permanent tissue. Simple permanent tissue is composed of similar type of cell. All the cells are same type, but in the complex permanent tissue, it is composed of different types of cell, but they are performing a common function, and that is called as complex permanent tissue. And these are not dividing cells. Division capacity is lost, and dividing cells in the plants, that is only on the meristematic part, and simple permanent tissue, they are performing the specialized differentiated function. And there are mainly three types of tissues are present on plant body. One is parenchyma, second is collenchyma, and the third is sclerenchyma. They are composed of similar type of tissue, and we will study about the characteristic feature of parenchyma, collenchyma, and also sclerenchyma. And complex permanent tissue, it is composed of different types of tissues, but they perform a common function. and they are also not dividing cells, and they are called as vascular tissue because they are the conductive tissue in the plant body. They are conducting water or water, mineral nutrients, and also the prepared photosynthetic product into different parts, and these are two types. One is xylem and the second is phloem. Xylem is conducting water and mineral nutrients, and phloem which conduct photosynthetic products or carbohydrate from leaf to different parts of the body. So this is the outline of plant tissues. Plant tissue can be divided into meristematic tissue and permanent tissue. And what are the characteristics of meristematic tissue? Meristematic tissues are present only on the growing part of the plant. The mechanized part or mechanical strength is not provided by the meristematic tissue. They are very smooth, and the cell wall is composed of only cellulose, and they are actively dividing cell with a large well-developed nucleus, abundant cytoplasm, and vacuoles are totally absent. If it is present, the vacuole must be smaller. And the meristematic tissue can be divided into three different types on the basis of position: apical meristem, intercalary meristem, and also lateral meristem. Apical meristem and intercalary meristems are primary meristem, and lateral meristem is a secondary meristem. Apical meristem is present in both monocot and dicot. Intercalary meristem is present only on monocots, and lateral meristem is present only on the dicots. And permanent tissues, it is performing a specialized function. They are non-dividing cells, and they are derived from the meristematic tissue due to the differentiation, and it is of two types. One is simple permanent tissue and complex permanent tissue. And simple permanent tissue, they are composed of similar type of cells, and simple permanent tissues are parenchyma, collenchyma, and sclerenchyma. Complex permanent tissue is composed of different types of cells but perform a common function, and these are the vascular tissue. And xylem and phloem are the complex permanent tissue that is present in the angiosperm. Next, we will discuss about the complex permanent tissue, and then the anatomy of the stem and the root, then about secondary thickening. So it is a simple chapter, as a brief chapter, and which may carry maximum of 3 questions. Normally 2 to 3 questions. In rarely 4 questions maybe came in the our Neet examination. So write the note about the primary meristem and meristem and permanent tissues, and we will move into the next part, the permanent tissues. Permanent tissue can be divided into simple permanent tissue and complex permanent tissue. Simple permanent tissues are differentiated tissues. They are performing specified function, and they are deriving from the meristematic tissue. And permanent tissue can be divided into, simple permanent tissue can be divided into 3 major types, which is present in the plant body: parenchyma, collenchyma, and sclerenchyma. Parenchyma, we know that simple permanent tissue, it is composed of similar type of cells, and parenchyma is a living tissue, living assimilatory tissue. They are non-mechanical, and they are present on major organs of the plant like leaf, root. The metabolic activities are done by the parenchyma tissue. And the cells are isodiametric. Cell wall are thin, composed of cellulose only. No any other chemicals like pectin, lignin, suberin, etc. is, or hemicellulose is absent. And cells are compactly arranged. With or without intercellular spaces. If intercellular space is present, it is very small. And parenchyma, which perform photosynthesis, and it acts as a for storage and also secretion. These metabolic activities, assimilatory activities, are done by the parenchyma. And parenchyma may be in some aquatic plants. The parenchyma combine to form certain air gap or air gap, and this type of parenchyma are called as aerenchyma. So in aquatic plants like Nilambium, lotus, etc., the plant body which have aerenchyma is with gas chambers, and it help in the buoyancy in water body. But in the leafy region, the parenchyma which have more chloroplast in their cell, the mesophyll cells, and this type of parenchyma cells with a large number of chloroplasts. These are chloroplasts, and this type of parenchyma with a large number of chloroplasts is called as chlorenchyma. Chloroplast containing parenchyma are called as chlorenchyma, and chlorenchyma is undertaking photosynthesis, and parenchyma is a living assimilatory tissue. This is living assimilatory tissue. And the second type is collenchyma. Collenchyma, which is present hypodermis region. And what is hypodermis? Just beneath the epidermis, especially in the stem region, that is the primary stem region, and also in the petiole. Just below the epidermis we can see collenchyma. And collenchyma are living mechanical tissue. They are living cell. Cytoplasm is present, protoplasm is present, but they are mechanical because their corners of the cell wall are thickened with pectin. Thickened with pectin. So this is a cell, this is another cell, this is another cell. So in this cell, the corner regions are thickened. So it provides mechanical support, it provides flexibility to the plant body, but they are performing their metabolism. So second type of tissues are collenchyma tissue. It is present on hypodermal region, and thickening are present on the corners by pectin. Intercellular spaces are absent because cells are compactly arranged. These are mechanical tissue, living mechanical tissue, and provide the mechanical support to the growing parts like young stem, petiole, etc. So two tissues, one is collenchyma and another is parenchyma. And the third is sclerenchyma. Sclerenchyma is highly lignified cell wall. The secondary cell wall is present. So if this is a cell, their internal part of the cell is totally lignified with a large amount of lignin. So the cytoplasm or protoplasm is absent. The lumen is very narrow. So it is a dead mechanical tissue. So narrow lumen. So the cell is totally dead and mechanically strong, and it is of two types. Fiber is somewhat elongated structure with the tapering ends, and the cell wall is highly thickened, and it is present in the xylem and phloem. Xylem fiber and xylem phloem, phloem fibers are examples for fiber sclerenchyma. And the second is sclerid. Sclerid is oval in shape or spherical, and it is highly thickened. The lumen is very small, and the cell wall is highly thickened with a lignin, and it is present in the fruit wall of nuts. This is very important. Fruit wall of nuts and a pulp of fruits like sapota and guava. That's why it has a gritty taste. And leaves of tea and seed coat of legume has a presence of sclerids. Sclerids are round sclerenchyma cells, and it is also present in the nut shell of the coconut. It has high thickened nature is due to the presence of sclerenchyma. So sclerenchyma is highly thick with a highly thickened cell wall, and it is dead mechanical tissue because lumen is absent. So this is about permanent tissue, simple permanent tissue. There are three types of simple permanent tissue: parenchyma, collenchyma, and sclerenchyma. And the next is complex permanent tissue. There are two types of complex permanent tissues, and the complex permanent tissues are vascular tissues. They are conducting water, mineral nutrients, and photosynthetic products. And there are two types of complex permanent tissue. One is xylem and another is phloem. Xylem, which transport water and mineral nutrients from root to upper part of plant. It is always unidirectional. That is always from root to upper part along the transpiration pool, and that we will study in the chapter transport in plants. And the xylem is composed of tracheids, vessels, xylem parenchyma and xylem sclerenchyma. Xylem components are tracheids, vessel, xylem parenchyma and xylem sclerenchyma. And In the case of angiosperm only, the vessels are present. In the case of angiosperm only, the vessels are present. So in gymnosperm, the vessels are absent. Vessels are absent in the case of vessels. So the components are tracheids, vessel, xylem parenchyma and xylem sclerenchyma. We know that here sclerenchyma is a dead tissue and all others are living tissue. Sorry, tracheids, vessels and xylem sclerenchyma are the dead tissue, but the xylem parenchyma is the only living tissue. Only living tissue. And the second complex tissue is phloem. And phloem, which transport organic food, that is a photosynthetic product or organic material from leaf to different parts of the body. So this is a leaf. If this is a leaf and this is a stem, so here it is transporting the food from leaf to different parts of the plant. So it is, the direction is a bidirectional direction. So the leaf synthesizes food material through photosynthesis, and that photosynthetic product which is transporting to different parts through phloem. And phloem is composed of sieve tube element, companion cell, phloem parenchyma and phloem sclerenchyma. Sieve tube element and companion cell. Sieve is just like the perforating region, and this is a sieve tube element. And sieve tube element is very important because sieve tube element is a enucleated mature cell present in plant body. Enucleated, that is, nucleus is absent in the sieve tube element, and it has cytoplasmic strands just like this. Cytoplasmic strands are present, and sieve tube elements which are devoid of nucleus, and sieve tubes and companion cell. Companion cell is always seen associated with the sieve tube element. They are closely associated because the functions of the sieve tubes elements are controlled by companion cell, because the nucleus of the companion sieve tube element is absent. No nucleus, but it is the major transporting element for the food material in the plant body. Sieve tube elements are the major transporting element in the plant body. And gymnosperm lack. So in the case of gymnosperm, sieve tube elements and companion cells are absent. Sieve tube elements and companion cells are absent in gymnosperm, and they have sieve cell, not a sieve tube cell, it is sieve cell and albuminous cells are present. Sieve cell and albuminous cell. So sieve tube element and companion cells are compactly arranged, so they are closely packed, and there is perforation, plasmodesmatal perforations are there. And the functions of the sieve tube elements are controlled by the nucleus of companion cell, and the transport of food material will be taken place through the phloem elements. And the next is phloem parenchyma. It is a parenchymatic tissue present along with phloem, and an important one, that is the monocote stem do not have phloem parenchyma. Monocote stem do not have phloem parenchyma. So parenchyma, phloem parenchyma is absent in monocote stem. And phloem sclerenchyma, it is a phloem fiber, or it is also called as bast fiber, and it is economically important fiber in we are using as jute fiber are the example for bast fiber. So here Xylem is the transporting water and mineral nutrients, and phloem which transport organic food material from one part to another. And on the basis of the arrangement of these vascular element, the vascular bundles or vascular tissue system can be of different types, like conjoined, collateral, open and closed vascular bundle. That is very important for the classification. And before that, in the case of xylem, there are two types of xylem. In the phloem is also present the two types of phloem, but xylem is very important and it is important for the classification. And there are two types of xylem. One is protoxylem. Protoxylem is first formed xylem. It is first formed xylem and it is smaller in size. First formed xylem, and it is smaller in size. And there are metaxylem. Metaxylem is later formed xylem. So the arrangement of metaxylem and protoxylem is very important. Here, when we are taking a vascular bundle, this is a metaxylem. It's with a larger vascular, it's a larger diameter, and this is the arrangement. So this is the epidermis and this is the pith region. So here, the protoxylem is towards the periphery. Protoxylem is towards the periphery and metaxylem is towards the center or pith. So protoxylem, this is protoxylem, smaller sized xylem, and metaxylem is larger sized. So when protoxylem is towards the periphery and protoxylem towards the periphery and metaxylem towards the center, that arrangement of xylem is called as exarch arrangement. Exo means outer side. Exarch xylem. And exarch xylem is a characteristic of root. Exarch xylem is a characteristic of root, because root is involved in absorption of water. So the protoxylem which absorbs small amount of water, that will be transferred into next one, and next one the larger one will translocate the water into upper part. So the effective absorption of water that require exarch arrangement. So exarch xylem is a characteristic feature of, it's a characteristic feature of all roots. Both the monocot and dicot root have exarch xylem. And in the case of stem, this is the periphery and this is the pith region. This is xylem arrangement. That is metaxylem towards the periphery and protoxylem towards the center. This is protoxylem. This is metaxylem. So this arrangement is called as endarch arrangement. Endarch. So when the protoxylem towards the center and metaxylem towards the periphery, that is called as endarch arrangement, and it is a characteristic of all stem. It is a characteristic of all stem. So vascular bundle and arrangement of xylem is very important. And on the basis of the arrangement of vascular bundles in a plant, it can be, there are different types of vascular bundle. First one, when we are taking a vascular bundle, this is a vascular bundle. Here there is xylem and here there is phloem. That is, when the xylem and phloem side by side, xylem and phloem side by side, xylem and phloem side by side, it is called as collateral vascular bundle, and it is in the same radius. Both the xylem and phloem are in same radius. Then it is called as conjoined vascular bundle. Conjoined. So conjoined collateral. What is conjoined vascular bundle? Conjoined vascular bundles are the xylem and phloem arranged on same radius, and when it is arranged on side by side, it is called as collateral vascular bundle, and it is a characteristic of all stem. In stem, there is conjoined collateral vascular bundle, and in conjoined collateral vascular bundle, so this is a conjoined collateral vascular bundle. here xylem is present, and in the lower part there is phloem is present, but in between xylem and phloem there is cambium is present. We know that cambium is a lateral meristem or secondary meristem. When the cambium is present, then it is called as conjoined collateral open vascular bundle. Secondary thickening is open. Because cambium is present, lateral growth or secondary growth is present, such a vascular bundle is called as conjoined collateral open vascular bundle, and it is a characteristic of dicot stem. It is a characteristic of dicot stem. So, in the case of stem, conjoined vascular bundle, conjoined collateral vascular bundles are present, and in the dicot stem there is in between xylem and phloem there is a strip of cambium, and that's why it is called as conjoined collateral open vascular bundle. And in the stem we know that there is another character, there is endark xylem. So this is the identification character of a dicot or a stem. In stem it is endark and it is conjoined collateral. When the cambium is present, it is dicot. If it is endark, it is a stem. If it is conjoined collateral, it is a stem. And when the cambium is present, it is a dicot stem. And when it is a vascular bundle, here phloem, xylem is present, and in the lower part there is phloem is present, but so this is xylem and this is phloem. So here phloem is xylem, here phloem and xylem, in between phloem and xylem there is a presence of cambium, then it is called as open vascular bundle. It is present side by side, it is called as conjoined collateral vascular bundle. And when there is phloem on the upper part and xylem on the lower part, and this is conjoined collateral vascular bundle, and the xylem is endark, but vascular cambium is present, so it is closed vascular bundle. Cambium is absent. When cambial strip is absent, then it is called as closed vascular bundle. Secondary thickening is closed. It cannot undertake secondary thickening, and it is a characteristic of monocot stem. And monocot stem we already studied. Phloem parenchyma is absent. So endark xylem. Endark xylem is a characteristic of stem, and it is with conjoined collateral open, conjoined collateral and closed vascular bundle. And in the case of roots, there is exarch xylem is present, and when we take the pith, the stellar region, this is the xylem with exarch arrangement. One xylem, second xylem, third xylem and fourth xylem. But phloem is present in between them, and xylem and phloem on different radii, and this arrangement is called as radial vascular bundle, and it is a characteristic of root. In all roots, exarch xylem and radial vascular bundle is a characteristic feature. The xylem and phloem which arranged on radii. So this is a basic idea about root, stem and the tissues. Next we are discussing another important part, that is called as heartwood, sapwood, etc. The secondary meristem or cambium, vascular cambium, is responsible for the secondary thickening in the dicot plants, and the activity of cambial activity is different in different season. So on the basis of the season, the cambial activity is different. In spring season, the cambial activity is very high. In spring season, cambial activity is very high and it produces large number of ciliary elements having vessels with a wider cavity. The vessels are with a wider cavity, and this wood is called as spring wood, that is formed during the spring season. We know that the activity of cambium is different in different season. In the spring season, the cambium is very active, actively dividing and produces large number of ciliary elements with wider cavitied vessels, and this is called as spring wood, or it is the early wood. And early wood is lighter in color and with a lower density. The diameter, the vessels are wider and it is lighter in color and lower density. But in the case of winter season, cambial activity is very less active. So it is very less active. So fewer ciliary elements are produced, narrow vessels are produced. Fewer ciliary elements and vessels with narrow lumen is produced, and this is called as autumn wood. So autumn wood is a resultant of winter cambial activity and it is called as later wood. And the autumn wood is dark in color and high density. Autumn wood is dark in color and with high density. So when we cut a tree, there are the concentric rings are present. The concentric ring. The first ring is with a darker color, other is with a lighter color. Again darker color, lighter color. This is due to the presence of in a year spring wood and autumn wood will be alternately arranged, and this is called as annual ring. Annual ring is the concentric arrangement of spring wood and autumn wood, or early wood and late wood in each year. So there is a branch of biology that deals with counting of the age of the tree. Counting the age of the tree on the basis of the annual ring. When we cut a tree, there are concentric ring. Two concentric ring. That is, one is for early wood, that is called as spring wood, and the second is the autumn wood. When we count these autumn wood and spring wood, that is the annual ring, we can calculate the age of tree, and that branch of biology that it deals with counting the age of tree by by counting the annual ring, and that is called as dendrochronology. So what is dendrochronology? Dendrochronology is calculating the age of tree by counting annual ring. So what is annual ring? The concentric rings due to the difference in the cambial activity in different season. That is, a annual ring that is equal to early wood plus late wood, or spring wood plus autumn wood. This is an annual ring. So in annual ring, two rings are present. So one ring is darker, another is lighter, then again darker, again lighter. So this is 1 year and this is 2 year. If there are 4 rings are present, it will be 2-year plant. So we can calculate the age of the tree by counting the annual ring, and that branch is called as dendrochronology. It is important. So what about the cambial activity? Cambial activity is different in different season. Cambium is a secondary meristem and it is present in dicot plants only. So only in dicot plants we can see the annual rings, not in monocot. When you cut a palm tree or date palm, any palm, we cannot get the annual ring. We can see the annual ring only in dicots like a teak or mango tree or a cashew. This type of dicot plants only we can see the presence of the annual ring. So we can calculate the age by counting the annual ring due to the difference in the cambial activity in different seasons. So this is about the brief description about the chapter anatomy of flowering plants, and it has more weightage in that group. Around 3 or 4 questions will be asked in the examination. So read the NCERT thoroughly and write all the notes what we have discussed in the class, and do maximum question related to the chapter that is from the previous question paper, and study well. Thank you.
Bright Education Center, Medical and Engineering Entrance Coaching Center, Hilal area, Doha, Qatar. For further enquiries, contact 306000679.